mechanism B and has been shown by a few studies to be positively selective. So here we're going to plot an ILDS scan, which is on the y-axis against genomic position. And this is what our scan looks like. We're applying this in windows, which is what each dot represents, and you can see these peaks. Then we call regions of this genome significant, which are colored in orange, and there you go. Our highest peak is TCDB. So we clearly are able to resolve known positive controls in known pathogens. So next, we're going to apply this to human gut commensals. And so this is a scan conducted in the gut commensal called Ruminococcus bromi. And what we pick up here is a sweep at this locus called MDXEF, which is involved in maltodextrin metabolism. What is maltodextrin? I had no clue. I had to read about it. But actually, it turns out that it's a very, very common additive added to highly processed foods. Like that salad dressing that you had, that goopy texture that it has, that we like so much, that is because it has maltodextrin. Or even baby formula, or sriracha. I encourage you to go to your fridge and check it out. You're going to find maltodextrin in it. And so this has been actually really commonly added to highly processed foods in the Western culture. If we take a look at the haplotype surrounding that locus, you can actually see a region of homogeneity right around that gene, resembling a gene-specific sweep. So what I'm showing here, each horizontal line is a different haplotype coming from a different host, and here you see that selective sweep. We next asked, is the sweep of MDXEF specific to the westernized communities? We looked at this dataset called UHGG, which comprises multiple publicly available datasets, and many of the communities are both from Western, colored in gray, and non-Western communities in red. And so here's the scan of Ruminococcus bromi. I'm showing a sample of our scans. On the top are non-Westernized populations, and on the bottom are Westernized populations, and colored here in yellow is the MDXEF locus. And it only shows up as significant in Westernized populations, not in non-Westernized populations. So it's possible, indeed, that our microbes are evolving adaptations in response to changes in our diet in our recent history, especially to these really, really important, strong additives that are added to highly processed foods. We can take a look at other species in our gut microbiota. And in fact, we see pervasive selective sweeps in multiple species. On average, we see around 5 or 10 peaks at selected sites per species. And so adaptation seems to have been a very common pervasive force shaping the genomic diversity of our gut microbes. Here are some of the scans. Again, other species show peaks at the maltodextrin metabolism gene. We see genes that are involved in the starch utilization systems, which are known to also evolve within hosts. And then we also see transcriptional regulators under evolution as well. And this is just a flavor of some of the categories under selection. And so evolution is actually important, and it's occurring commonly not only within us on short timescales of days and months, likely mediated by horizontal gene transfer, but also it's pervasive and happening across us through migration and recombination. Maybe you'll pick up a souvenir from this conference and take it home. So with that, I'd like to acknowledge some of the very close co-authors on this work that I presented, as well as my funding. I'm recruiting postdocs and graduate students, so please come and meet us. This is my research group outside of my lab at UCLA. And happy to take any questions, and thank you for coming.